Hello, my dear students. Today we will pass on to the next chapter, chapter 6, Genes and Chromosomes. Here we are discussing about the linkage and recombination. Already we studied about the pedigree analysis. So what is a genetic linkage? All of you know that the chromosome theory of inheritance. So, in the George John Mendel, assume the characteristics are determined by certain factors of inheritance which are able and particulate and segregate independently at the time of gamete formation. So, after the rediscovery of Mendel's law in 19,000, Walters et Sutton, an American graduate student, and Theodore Boveri, a German biologist, in 1903 have independently observed a parallelism between the behavior of chromosomes and Mendelian characteristics, and they noticed that the several similarities between the two. And so the Sutton and Boveri postulate the chromosome theory of inheritance. This theory states that Mendelian factors, the genes, are located on chromosomes and is the chromosomes that segregates and assorts independently during meiotic cell division. A simplified version of the chromosomal theory is presented the figures, and that is present in your page number 191. And so the two duplicated and paired homologous chromosomes in a tetrad assembled to the metaphase stage of the meiosis 1 can separate in two possible ways. And this lead to independent assortment of the chromosome and the genes they carry in four possible ways. After meiosis 2, four types of gametes are formed, and these are separated in the F2 generation. The 9:3:3:1 ratio is forming. Okay, and then pass on to the genetic linkage. The discovery of linkage. William Bateson and R.C. Punnett were working with several traits in sweet peas, a gene for purple and red flowers, a gene for long pollen grains and round pollen grains. What is genetic linkage? Genetic linkage is a sequence of DNA sequences that are close together on the chromosome to be inherited together during the meiosis phase of sexual reproduction, or the tendency of genes to remain together in their original combination during inheritance is called the linkage. So, in the case of linkage, the group of genes which behaves as a unit is said to be linked, and the phenomena is known as linkage group. The number of linkage groups in an organism is the same as the haploid number of chromosomes or the number of pairs of chromosomes. So, the tendency of two or more genes of the same chromosome to remain together during the process of inheritance that we called as to the linkage. According to T.H. Morgan, the degree or intensity which two genes are linked together is known as linkage value. Then why genetic linkage? Because in organism genes are far more in number than the chromosomes, which implies that each chromosome contains many loci, many position. Loci means the position of the chromosome. Genes far apart on the same assort independently are not linked. The position of the gene, that is locus, occurs in the prophase of the meiosis 1, where homologous chromosomes break at identical locations and rejoin with each other. Two genes are said to be under linkage or linked when they are located on the same chromosome. Example P is T tall, T short, R red and R white. When and who discovered linkage? Shortly after Mendel's work was rediscovered, exceptions to this rule were found. In 1906, the British genetist William Bateson and Regard Punnett rediscovered the phenomenon of linkage in sweet pea. Can linked genes ever separate? Yes, they can through the recombination process. Factors influencing crossing over and linkage. So the linkage is divided into two types, the complete linkage and incomplete linkage. And then factors that influencing linkage. First one, the distance. The strength of linkage is inversely proportional to the distance between the linked genes. Then the age. Increasing age, the chance of crossing over are reduced and hence the linkage increases. Then X-rays. Exposure to the X-rays reduces the strength of linkage. Then fourth, temperature. Increase in temperature decreases the strength of linkage as chance of recombination increases. Then the sex. The male Drosophila shows little crossing over. The phenomena of crossing over is quite common in the female fly. Then X-rays, the temperature and chemicals. These are some of the factors that affect linkage. Then types of linkage. Two types mainly, complete linkage and incomplete linkage. The complete linkage is exhibited when the genes for a particular character are present very close to one another. It is due to non-break in the gene combination situated on the chromosomes. It is very rare in nature. The most example of complete linkage in male Drosophila for grey body color and long wings. The Morgan demonstrated through an experiment and proved that the complete linkage is occurs into the animal. Do not get separated, very close. Example in Drosophila, the 9:3:3:1 ratio we get. And so the Morgan demonstrated through his experiment on Drosophila that crossed between the male with grey body and long wings and female with two recessive characters. Black body and vestigial wings results in F1 hybrid which are like the male present. If a male from F1 generation is back crossed with to the double recessive black vestigial female. 4 kinds of offsprings in equal numbers as the result of independent assortment are expected. There are, however, only 2 types of offsprings which resemble the two grandparents, and this suggests that the genes for grey body and long wings are linked together and thus show complete linkage. Then the incomplete linkage occurs when the genes for different characters are separated at the time the gametes formation due to breaking and exchange of chromosome pieces during meiosis can separate because distance is more. Recombination due to the crossing over. Then the recombination. And then what is a crossing over? Crossing over means the mutual exchange of blocks or homologous genes between a pair of homologous chromosomes is known as crossing over. Because in the crossing over, the homologous pair of chromosomes, they exchange their parts. The mechanism of crossing over involves breaking and rejoining the chromosomes in a synaptomial complex. The cross with the two linked genes, how it crossed with two linked genes. And what is recombination? What is recombination? So we studied this recombination in the next